All right, guys, we're looking at more of the ways that microbes avoid the immune response. And we already looked at some of the ways that microbes avoided phagocytosis. Now we're looking at more than just that, all right? One of the aspects that we need to talk about is tolerance, all right? This is a case where the immune system basically puts up with, tolerates, right, the pathogen's presence. And there are a lot of reasons why the immune system would accept the presence of an antigen or antigens on a pathogen. One example would be fetal exposure. Early on, when the immune system is training, right, those B cells and T cells are training as to what is self, what is non-self. Fetal exposure makes it very hard for the immune system to assign what is self and what is non-self. There are these mistakes made where the immune system comes to accept the presence of an antigen that may be a foreign antigen because it is mistakenly taking that as a self-antigen. Okay? Another reason would be just high exposure, exposure to high concentrations of a particular foreign antigen. So if it happens to be in the system and it's always there, it kind of makes sense that the immune system would come to accept that that must be a normal thing. Okay, and another one that I think is really interesting is molecular mimics. The fact that pathogens' proteins, their antigens, look a lot like the proteins that are in our own system. Now, these organisms don't have a brain, right? They're not thinking about what it is they are going to have on their surface. They just have genes. They express those genes. Some of those proteins are on the surface. But it does make sense that if some of those antigens look just like our own, aren't they going to fare better as a pathogen? They're going to go unnoticed as a pathogen because these antigens look pretty much like the normal host pathogen, normal host antigens. Okay, so pretty interesting. And one that is a molecular mimic that you may know about is the proteins that are found on strep A organisms. So people have strep throat. They have these antigens that are on their surface. And the immune system has a choice to make, right? Sometimes the immune system will come to tolerate the fact that it's there, and then we risk the infection remaining, or they go the other way, right? They go the other way and they go after those antigens and after the very same proteins, and in this case it would be rheumatoid arthritis, right? So they can come to tolerate it, which would leave the joints alone, but the infection might get the best of us, or they go after it, and then we have an autoimmune condition. All right, another really interesting one is antigenic disguise. So imagine this, right? You show up as a pathogen, but you dress up, right? You dress up to look like just another host molecule, all right? Or part of the host. So one example would be protein A. Protein A exists on the surface of some cells, like Staph aureus, for example. And I have a little picture here of what it might look like. So here's an organism, a circular organism like Staphylococcus. And on its surface, you might have this protein A, they call it. Now, protein A is known to bind to the constant end of antibodies. All right, so here comes an antibody, right? The variable portions of this antibody are out here, right? So these are the variable portions, and they're going to stick to some foreign antigen, I suppose, right? But here we have a cell we'd rather get rid of, the Staph aureus bacterium, but it's not being bound by the variable portion, right? It's being bound by the constant portion and another. And this would be covered with these antibodies, which usually is a bad thing for a pathogen, right? They're tagged for destruction. But in this case, the constant end is bound to the organism and the variable portions are pointing out. This is not a flag for the immune system. This is just another antibody, and so this organism goes unchecked, right? It's not going to be bothered by the immune system. Okay, another form of antigenic disguise. Host molecule binding. So, and one organism that does this is Treponema pallidum, which is a syphilis organism. Okay, so let's take this organism, and this happens to have receptors on it, like you see here. And each of these receptors, imagine that each of these receptors binds to, I don't know, pieces of our own system, like pieces of collagen in the dense connective tissues, or fibronectin, which is another connective tissue. So if these bind up, let's say this binds up a normal dense, some kind of fiber that's normally in our connective tissues, right? And it's all over this bacterium like that. Well then, some kind of cell that comes along and looks at this, let's say it's a macrophage in the area, it's not going to see that there's a cell there. It's going to see that there's fibronectin or collagen there. Okay, some kind of host molecule. Again, it's disguising itself. And one last one would be a cell-bound coagulase, like group A strep organisms. They can do this. So imagine you've got a cell and it's got an enzyme on its surface, and in this case it forms clots. Okay, so imagine you've got clots forming, right? And that would happen with fibronectin, right? So coagulase is going to form these fibrin nets all around the cell, right? Now, you might think, well, that's not good for the cell, right? Clots are going to be a way of keeping the cell from moving about. And you'd be right. That's one of the things we learned, right? But some of these organisms that form clots also can break the clots down when they need to. Okay, so they have the ability to form clots and break them down so they can move around. But this is still going to disguise and hide that organism. Okay, so coagulase, another antigenic disguise. All right, another way that an organism could go unchecked and avoid the immune system would be antigenic variation. So these bacteria more than likely have antigens that are foreign, that our immune system is usually pretty good at identifying. But it also remains a fact that these organisms do evolve faster than we do, and which basically means their DNA is changing more quickly for various reasons, and if the DNA changes, the proteins and their shapes change. Okay, so it's harder and harder for the immune system to take notice of this particular foreign antigen, but that foreign antigen is always a moving target, right? But if because the genetic change is there, right? So genetics are changing, causing these antigens to change on the surface. Neisseria has fimbriae proteins that do this all the time, all right? The flu has viral proteins that do this all the time. And these are just a couple of a zillion examples we could go over. We could go with the coronavirus, same deal, okay? They think that a vaccine that we come up with, as soon as next year may only be effective for a few months because the coronavirus is also constantly going through antigenic variation, okay? So it's not a guarantee that getting that vaccine is going to protect you for years to come. All right, so let's take the flu because it's a most common one. It's an easy one to take and talk about. So influenza A, viral proteins. Here we have a bunch of them. Okay. Now again, and for this, I don't expect you to memorize each one of these, but they all have names, right? They have the H protein. They have an N protein. And some of them are for attachment and penetrating into the cell, right? So they're going to actually attach and infect their host cell this way, just like the spike protein did or does for coronavirus. But we have a bunch of these on the surface. And these are always subject to change. We've got the H and N. Sometimes you'll hear H1 or H1N1, right? So basically the name of the flu virus, the particular strain or variety, we're using the proteins on the surface to give it a name. And some of these are more conserved. In other words, they don't change very much at all, or else it wouldn't, the virus wouldn't be successful. And some of them are subject to change quite frequently. The question becomes, if antigenic variation is about change, and these are the ones that will change, H and N, okay, what causes that change? Okay, what causes the change to the genetic material within that virus? One thing that leads to this change would be something called genetic drift, a random mutation. Now again, I think random and I think not very accurate. If you spin me around, right, with a dart and then tell me to hit the dartboard, maybe I'll hit the dartboard. Maybe I'll hit somebody else, right? That would be a really negative effect. But what are the chances I'm going to hit a bullseye and I'm actually going to have a positive result? Not great. So genetic drift and random mutation can result in beneficial results, but it's a very slow process because it's hit or miss, okay? These DNA code errors can happen because during replication, right, the DNA polymerase makes errors, or the change due to other factors, right? You could have chemical mutagens or radiation, any variety of things. So if we're talking about antigenic variation being a problem for the human immune system, it absolutely is, but most of the time genetic drift is not going to be the main driver of that, though it is one. One of the main drivers of that antigenic change that will be problematic will be a mixing and matching, a reassortment of whole genes between viruses, or the recombination event, where whole genes are swapping back and forth between viral genetic chromosomes. And this is going to occur relatively quickly, all right? Whole genetic chromosomes could be swapped, okay? And the example that we see here for this kind of change might occur between, let's say, a bird and humans, right? There are plenty of examples of bird flus, swine flus, bovine flus, and they started in this other animal, right? A zoonotic source, okay? A lot of times when you think about an animal that is a source like that, we call it a reservoir. It's normally in that animal, perhaps. But what can happen is for humans, let's say they're humans, people that raise chickens, right? And they're in close proximity to chickens all the time. The chickens have some kind of bird flu that normally attaches and only infects them. But then by some rare chance, the human also becomes infected with that flu virus from the bird, okay? So a non-human virus with certain genes that make this green attachment protein, let's say, right? That's what allows it to infect the bird. But then the same person, let's say, has acquired a human virus, some other flu. And of course, this other color here allows it to infect humans. But if the person has both viruses, it is possible for a repackaging of the viruses when they infect and then lyse the cell to have both. Okay? And now it's got a whole new host. It's got a whole new world, right, to infect. And this is what we usually worry about when it comes to viruses like this. They jump between species, just like coronavirus went from a bat to possibly some other species and then to humans. Or maybe it went straight. We don't know completely if it went straight from bat to a human. But this is a pretty common thing and something we need to have respect for and understanding of. All right. One last problem for the immune system are the host of secretions, what the bacteria are able to secrete into the system. Sometimes it's not the bacterial load, that's a strange term, or microbial load, that would indicate, okay, what is the sheer quantity of bacteria that are in my system causing the infection? The sheer number of bacteria could be the problem, and we're reeling, our immune system's having trouble because of that. And we could knock down that bacterial number with antibiotics maybe. But sometimes the microbial load, the sheer number of bacteria causing the infection, can be very small, but they secrete tons of various chemicals and molecules that cause more problem than the sheer number of bacteria. One example of a secretion that bacteria will produce would be enzymes. Enzymes that are useful for invasion and the spread of that bacterium in the human system. And some of them might include hyaluronidase, collagenase, elastase. We happen to have hyaluronic acid, collagen, and elastic fibers in our system. They're all part of connective tissues. They're everywhere. And in order to be able to move around, invade, and spread throughout our system, these bacteria produce enzymes like these to chop down. It's like hacking through, a really dense forest to get somewhere, right? Clostridium will be a good example, like C. diff. That's the one on the tip of my tongue. There's other Clostridia. There's botulinum, right, which causes botulism. But C. diff is the big one that we think about nowadays. Streptokinase and staphylokinase. All right, we talked about how it was that cells could cloak themselves, right, and disguise themselves with a clot. Sounds like a bad deal for a bacterium, but they can also break free of those clots. Streptokinase, staphylokinase are enzymes that allow an organism to break the clot and run free. Okay, so when needed, they can form the clot, stay unnoticed from the immune system, and then when they need to, they can break down the clots. A lot of those organisms will contain both a coagulase for making the clot and maybe a strepto or staphylokinase for breaking free. Some of these organisms can produce literal antibody proteases, right? Antibodies are a protein, and some bacteria make proteases, enzymes that break those down. So that's pretty obvious how it's going to help the organism, right? It's going to be able to actually just cut down antibodies, not be affected by them. Pseudomonas is one of those. All right, one last bacterial secretion, and that would be the whole group of toxins. We have two big categories, endotoxin and exotoxins. Now you might think endo for within, right? And you'd be correct. And exo for without, outside. And it would lead you probably to a pretty close to the right understanding. But endo is a little misleading. Endo would refer to any molecules that are associated with the bacterium, but they're usually not in the cell, they're on the cell. So it's a slight variation of the use of the word endo, okay? Endotoxins are associated usually with the envelope of the cell, the outside surface of the cell. Otherwise, the immune system wouldn't bother with it at all, right, if it's inside the cell. But endotoxins would include some of the chemicals like we've talked about already, like LPS, right, lipopolysaccharide, from gram negatives on that outer membrane. They would be one example of an endotoxin. And when they're released, they can cause major inflammation, fever, coagulation, so clotting throughout the system, inside the blood circulation, not good, and anaphylaxis, so shock. So our immune system responds to all these chemicals like a bee sting, right? You're basically responding to these endotoxins when they're busted up and released out into the bloodstream like you would a bee sting if you were really allergic. Some examples of organisms that make these kinds of things: Salmonella, gonorrhea, right? Neisseria is the genus for that. Proteus. And then we have this other category of exotoxins, which are far more potent and problematic typically. They're more likely to cause the actual disease you're suffering from. Not the bacteria eating your tissues up, right? Causing damage there. The disease is the problem with the toxins themselves. So these are the organisms that just churn out toxins. They're released, right? They're not on the surface. They're released in the millions out into the bloodstream perhaps. And this definitely would cause possible shock and death if it's not dealt with quickly, okay? Some examples of places, organisms that produce exotoxins. We've got, again, Clostridium with its botulinum toxins, tetanus. You've heard of that, right? So these are neurotoxins, the tetanus toxins, and they are more of the problem than the organism itself. We got gas gangrene, diphtheria, cholera. These are pretty nasty sounding organisms. You've heard of them, I'm sure. All right, so we're going to call it quits now on some of these nasty aspects of pathogens. And I may have one more clip for you guys and at least a goodbye. I know we've got a lot left to do. So don't forget, I'm here for you. If you need help, absolutely, let me know. I'm emailing people back, but I'd be happy to meet with you online, okay? So I'll see you very soon with another clip.